A DNA is one of the possible double helical structures which DNA can adopt. A DNA is thought to be one of three biologically active double helical structures along with bDNA and zDNA. It is a right-handed double helix fairly similar to the more common bDNA form, but with a shorter, more compact helical structure whose base pairs are not perpendicular to the helix axis as in bDNA. It was discovered by Rosalind Franklin, who also named the A and B forms. She showed that DNA is driven into the A form when under dehydrating conditions. Such conditions are commonly used to form crystals, and many DNA crystal structures are in the A form. The same helical conformation occurs in double-stranded RNAs, and in DNA-RNA hybrid double helices. Like the more common bDNA, a DNA is a right-handed double helix with major and minor grooves. However, as shown in the comparison table below, there is a slight increase in the number of base pairs per turn. This results in a smaller twist angle, and smaller rise per base pair, so that aDNA 20 to 25% shorter than bDNA. The major groove of aDNA is deep and narrow, while the minor groove is wide and shallow. A DNA is broader and more compressed along its axis than bDNA. The identifiable characteristic of aDNA X-ray crystallography is the hole in the center. The gap in the center of the DNA structure is formed by the stacking of the base pairs, or sugar pucker. A DNA has a C3 endopucker, which denotes the base's proximity to the phosphate in the backbone of DNA. Comparison geometries of the most common DNA forms.